How can immune cell and gene therapies be manufactured at scale? Imagine turning tiny cells into powerful medicines that fight diseases like cancer. That's what scientists aim to do with immune cell and gene therapies. But making these treatments in large amounts is a complex puzzle. Let's break down how it's done. First, the process starts with collecting immune cells, usually T cells, from patients or donors. This is done through a procedure called leukapheresis, which separates blood components and isolates the immune cells needed. These cells can be processed immediately or frozen for later use and transported to specialized manufacturing facilities. Once at the facility, the cells go through purification to remove unwanted cell types and enrich for the target T cells. These are then activated using specific signals to prepare them for modification. Next, the cells are genetically altered to express therapeutic genes. Viral vectors have been used traditionally, but now non-viral methods like electroporation are gaining ground. Electroporation uses electrical pulses to introduce genetic material directly into the cells, making it faster and less expensive. After modification, the cells are expanded in controlled environments with cytokines, which are special proteins that help cells grow. This expansion can produce tens of billions of cells, enough for multiple doses. Once enough cells are grown, they are formulated with cryopreservation agents and frozen for storage and shipment. Quality control checks are then performed to ensure safety, potency, and purity before the therapy reaches the patient. Scaling up manufacturing involves automation and closed-system technologies. Machines like the Clinimax Prodigy can process multiple batches simultaneously with minimal human intervention, reducing contamination risks and lowering labor costs. For autologous therapies, which are made from each patient's own cells, production must be personalized, making large-scale manufacturing more challenging. On the other hand, allogeneic therapies made from donor cells can be produced in larger batches and distributed widely. Innovations such as improved cell isolation methods, faster gene delivery systems capable of processing billions of cells per hour, and microbubble-based separation techniques are helping overcome current hurdles. These advances make it possible to produce immune cell and gene therapies more efficiently, at lower costs, and in shorter time frames. This means more patients can benefit from these powerful treatments. From the immune system's point of view, this manufacturing process is about harnessing and enhancing natural defenses by reprogramming immune cells to better recognize and attack diseases. The challenge remains in producing these cells consistently and in sufficient quantities to meet global demand. But with ongoing research and technological improvements, the future of immune therapies looks brighter, promising more effective and accessible treatments for many illnesses.